Excellent. So, last speaker before lunch, MG, is going to talk about the adaptive evolution of expression in melanoma. Take it away.
Thank you. Yeah, so I'm MG Hirsch. I refined the title here a little bit from what's in the program, just to be a little clearer. So, talking about adaptive evolution of gene expression. So, we're good on this. So the different subclones within a tumor are going to have the different mutational profiles, but also the different gene expression and different phenotypic attributes. And so what we're interested in this project is how are these adapting to the environment? How are the different subclones adapting to either the same selective pressures from the environment, maybe like immune response, or maybe there are different selective pressures on different subclones that are making these subclones change in different ways. And so measuring things at the actual functional level is very difficult, so we're looking at gene expression, because obviously these selective attributes aren't acting directly on the DNA, they're acting on this functional level. So let's go through the data that was generated for this, and this was from Cenk's lab and Salin. So from an M4 mouse model, 24 single cells were extracted, isolated and cultured. And so these, we're calling them sublines. They kind of represent subclones, but within each subline, the cells are much more homogeneous than within an actual subclone. So each of these undergoes single cell RNA-seq. So we have gene expression by subline for each, gene expression for each subline. It also undergoes bulk whole exome sequencing, and we use Tricycle, which was developed in Cenk's lab. That's going to give us that mutation-based tumor tree that's been talked about some in the previous sessions. Each of these sublines was also implanted into a mouse individually, and like in vivo growth information was recorded. So we have information about how aggressive these sublines are. So for each tree, we can label these sublines based on these phenotypic attributes and kind of divide different groups of the tree. We also have, based on these mutational profiles, we can compare each subline and each group of sublines mutational profiles to those of human melanoma with known immunotherapy drug response. And so we can get pretty confident predictions about how these sublines are going to react to immunotherapy, and then a subset of that was validated experimentally, so we can say pretty confidently what these responses will be. So obviously that was just a toy example. This is the actual mutation tree that we're using. And we have this, colors are dividing up this tree based on these phenotypic attributes. So this blue group contains the 4 most aggressive sublines. They grow very, very quickly, and they don't respond to treatment. Then yellow, we're looking at sublines that still grow very quickly but do respond to treatment. And in the green, we have kind of heterogeneous growth, but generally lower growth rates, and again, respond to treatment. So we're looking at these 3 groups of the tree specifically because they have this range of phenotypes that we can compare. So that's kind of our data. What are we doing actually computationally? How are we determining this adaptation? So with these 3 types of data, the tree divided into these phenotypic features that we saw, as well as the gene expression, we're going to fit different models of evolution. So the first model, kind of our null hypothesis, is this neutral evolution, and we model that with Brownian motion. And this is kind of the case where the expression for a single gene is just changing randomly throughout the subclones. The second method that we fit is we are modeling constrained evolution, and this is with an Ornstein-Uhlenbeck process with a single optimum. And so this is the case where all of the sublines are kind of moving to a single expression level. And so when I say optimum, I mean there's some sort of one gene expression level that seems to be really good for these subclones. So all of the subclones are moving towards this one expression level. And the third option we fit is adaptive evolution, and this is, again, an OU process, but with 2 optima, and this would be the case where one group of sublines is moving towards one kind of optimum gene expression level, but a different group is moving to a different value. And so this would be the case, maybe there are 2 different selective pressures acting on the different groups of subclones, causing these different values, or it could be one pressure, but they're adapting in different ways. Then we perform a likelihood ratio test between these fits, and that's going to give us these constrained and adaptive genes. And this was done using Evogenics, which is a tool developed by Soumitra from Teresa Przytycka's lab at the NIH. So just a really quick overview of some of the results. So we did overrepresentation analysis of these adaptive genes with KEGG terms, and then here we've grouped these terms into larger groups just to give a really a bigger overview so that it's easier to see these trends. And then we have these adaptive genes separated both by the different groups of sublines that we considered, but also whether they're upwardly adaptive, meaning that that specific group of sublines is moving towards a gene expression level that's higher than in the rest of the tree, or downwardly adaptive, meaning that group is moving towards a lower gene expression level. And so if we start with the aggressive non-responder sublines, they're adapted to have a higher gene expression level in things like G protein coupled receptor signaling, membrane receptors, PI3K signaling, things that are related to cellular communication. And they're downwardly adaptive for things like mitochondrial and protein and ribosomal processing that are more related to cell growth. Whereas if we compare this to the aggressive responder sublines, we see almost the exact opposite trend, right? They're upwardly adaptive in cell growth and downwardly adaptive in cellular communication. So and then the non-aggressive responder sublines are kind of in between, a little bit heterogeneous, kind of reflecting the heterogeneous nature of their growth rates. So this really suggests that we have this really different adaptive evolution happening between these two groups of sublines. And so this could be that maybe there are two different selective pressures, and so they're adapting differently in that way. Or it could be one selective pressure, and they're just dealing with it differently. So one of the hypotheses is that these non-responders are dealing with this, like maybe the natural immune response, by modifying the gene expression of the genes that will cause immune escape, whereas this aggressive responder subline group might be just growing very quickly, so it doesn't really care if the immune response kills cells because it's just replacing them quickly. And so this kind of reflects the two different general subtypes of cancer that we see. These non-responder maybe slower growing, and then these quicker growing responder groups. So a few quick takeaways. Gene expression can be really helpful, specifically in this adaptivity analysis, because it's closer to that functional level that is actually being modulated by the selective pressures. We can identify maybe some of these functional pathways that give insight, and maybe even identify potential immunoresponse targets. Obviously, I didn't do this work by myself. So Erin, Molloy, and Teresa Przytycka are my current advisors at the University of Maryland and the National Library of Medicine at the National Institutes of Health. I've also been working a lot with Soumitra and then Qiping Day and Cenk who've been at the National Cancer Institute, who's been really helpful with supplying the data as well as helping with the biological insights. I'll have a poster tomorrow. It's number 22, obviously, in 8 minutes. I could only do a very small subset of what we've done. We've done a lot of validation on this, so please come talk to me if you're interested.
Question from Martin?
Really interesting. So how many, maybe you said it, but I missed this information, how many genes can you confidently assign to this?
That's a great question. Yeah, so we're looking at about 8,300 genes, about 8,000 genes total. And we really, depending on the subgroup, we're getting at least 60%, but sometimes closer to even 80% that we're really confident in. And then there is a group that's like kind of maybe, and then some of them where the estimations are just absolutely wild and we just throw them away.
Sorry, 80% of, so you consider it to detect?
We look at all of the genes, and then Evogenics, our method to assign adaptation, we'll get about 80% that we're very confident in these.
And are these genes, sorry, just one second. Are these genes, these are genes that you also find eventually expressed in
Yeah. Actually, let me clarify the last question because I may not have gotten it. There's about 9% that we label as adaptive. of all of them, we're able to say about 80%, yes, we think we're confident in what we're signing. The differentially expressed, we've looked a lot at that, and there's a lot of overlap, but there's also a lot that's different. And if we do simulation studies with this, we're seeing that we have a lot fewer false positives, which is really great, right? That's good news. But the false negative rate is very, very similar.
So if I get it right, you have a stochastic differential equation for the Uhlenbeck process,
Yes.
right? And that is this 8,000 dimensional space,
Yes.
right?
Well, we test each gene individually.
Yeah, right.
Yeah.
So in the example, it was very clear that one thing was going up and they were bifurcating. What does it happen in 8,000 dimensions? Is it easy to find this?
So we do each gene individually. So we have, so we're checking, is one gene adaptive at once? So it's not 8,000 dimensions. It's going to be the dimensions of the number of cells by the number of sublines.
But do you fit to the steady state, or because you don't have time series, right? Or you do have time series?
We do not have time
Yeah,
series.
So you fit the steady state of the OU process to this, right?
So we do a maximum likelihood to predict the,
and the steady state is a Gaussian, right, of the OU process.
no, I think is the answer. I don't know that I'm
The answer is
understanding.
yes.
Oh,
the
okay.
steady state of
Then
the
I
OU
don't
process
understand the
is a
question, I'm
Gaussian
sorry.
distribution, so you basically fit the mean and a variance.
So we also use replicates, so we have the mean and the variance of the
Right.
replicates of each subline, and then
So
we're
you compare
fitting.
a Gaussian against a
It's
mixture.
well, the OU is not entirely Gaussian, so it's the difference between the optimum value that we're estimating minus the current value of the gene expression, and then that has a weight, and then we add to that this Brownian motion of the within species or within subline variance, and that has a weight, and then that's summed.
All right. Well, I'll come to poster 20.
Yes, and I have the equations on there.
Oh, so
Last question.
yeah, thank you. So the most fascinating thing about it to me, is figuring out which genes go into which evolutionary mode. And so if I listen to your answer to the other question, about 800 genes were adaptive or so. How many of them were adaptive and had one?
So we
optimum,
had
and how many had two optimum?
yeah. So okay, so we have 8,000 genes that we're testing. Between like 7 to 12% of those were labeled as adaptive.
Okay.
In total, we only had 13 genes that were labeled as constrained. And then all of those, when we further tested to adaptivity, they could also be further redefined as being adaptive.
So I might have forgotten the names of things, but the constrained was the one where they only have one optimum?
Yes, yeah. Yeah, so constrained is where all of them are going to one optimum, and adaptive is
So
like
almost
this.
all of them are adaptive?
Yes,
Okay.
all of the ones that we identify
Okay.
as being either constrained or adaptive.
Most
Are
almost
adaptive.
the vast majority.
Yeah,
adaptive.
yeah.
Okay.
Very last, and then we have lunch.
Okay, I think I do not know the evolution 101 actually. It's not
You're
my
good.
domain. So is subline and subclone are same thing?
The sublines represent a subclone, so there's single cells extracted from a heterogeneous tumor that probably come from different subclones. But then the sublines, a single cell was extracted and then replicates. So within a subline, it's very homogeneous from that single cell, whereas a subclone would still have some heterogeneity.
Subclones are branches of this phylogeny that she showed. They can contain multiple subclones.
Yeah.
Okay.
Yeah.
Okay, thank you. So you can isolate experimentally those from gene expression.
Say it again.
I see you are isolating the 5 subline. So you can isolate those sublines using gene expression.
So these are isolated by extracting single cells from a
Oh,
tumor.
experiment.
Yeah.
Lab experiment.
Yes, yeah.
Okay, thanks a lot, MG, and thanks everyone.